Okay, so good afternoon. The light is very strong, so I cannot see much from the audience, but I will do my best. So I'm going to talk with you about a neurogenetic syndrome named Williams syndrome. And then I'm going to describe a therapy that was developed for a mice model with Williams syndrome by a colleague of mine, Professor Boaz Barak. And lastly, I'm going to describe to you the design of the study in which we are trying to translate the findings in the mice to humans with Williams syndrome. I'm a child psychiatrist. I'm located at Sheba. I've been treating and studying individuals with 2 neurogenetic syndromes for over 2 decades. One of them being 22q deletion syndrome, which is maybe less known than Williams, but much more common because it's 1 to 2,000. And Williams syndrome, we actually are the center for treating and study Williams syndrome in Israel. When I'm trying to explain what are behavioral phenotypes, I'm always giving these examples showing how genetic defect affects behavior in a simple test that I call, that I named the Ninette test. So Ninette, this is a picture of Ninette from about 2006. She was very popular at that time. And in the clinic, we do days for fun for the families, and we invited Ninette. And what you could see, that the event was both for Williams and for 22q individuals. And on stage, you can see mainly or only individuals with Williams syndrome because they are very outgoing. They like to sing and dance and very people-oriented, especially to strangers. So they are on stage. They are in it. But on the other hand, individuals with 22q, they are very shy, we say with social anxiety. So they will never go on stage to sing and dance with Ninette. So you see that a microdeletion on chromosome 7, which is Williams syndrome, causes a certain behavior in such a social event. And on the other hand, 22q deletion make the opposite. So let me tell you a little bit about Williams. It's 1 to 20,000 live birth. It is caused by a microdeletion on the long arm of chromosome 7. The phenotype is a mixture of medical diseases, intellectual disability, and very specific, very unique social behavior. We know that the microdeletion size is 2 megabase, 2 million nucleotides. Within the deletion region, there are more than 20 genes. We know for sure that one of them, the elastin gene, is responsible for the blood vessels problems. We are not certain yet which of the genes is responsible for the brain phenotype. These are the variety of medical problems these individuals are facing. I must say that it's a child psychiatrist or psychiatrist that treat them because the main issues are the learning disabilities and the psychiatric phenotype, but they also have variety of medical conditions, but not so severe. They have some stenosis of the blood vessels, most famous supravalvular aortic stenosis. They also have scoliosis. They are famous for having hypercalcemia, etc. I think this example of a patient of mine demonstrates nicely the typical behavior of a young adult with Williams syndrome. So Ido, at that time, was 30 years old. He entered my office. I was sitting there with a resident named Shimrit, and he asked me, please give me a piece of paper. And he wrote a song in Hebrew, which I translated. Song for Shimrit, you are very beautiful, you have a magnificent voice, and a smile that is very captivating. It was very nice to have met you with love and friendship. I love you, Ido. And so that was the song that he wrote. And on the other side, you can see what he drew here. Can you guess what it is? Heart. It looks more like avatiach, like a watermelon, but this is a heart. What it means that Ido is very affective in his relation to a stranger doctor, maybe inappropriately affectionate. And on the other hand, you can see that the drawing of the heart is like of a child aged 3 to maybe, like 3 years old. So in Williams syndrome, we have a gap between the verbal ability and especially the ability to express emotion versus the low visual, spatial abilities in mathematics. So they have peaks and valleys in their cognitive abilities. So far, there is individuals with Williams, they have high rates of ADHD, high rates of anxiety, not social anxiety, but worries, and they are afraid of noise, phobias, etc. But so far, we are like with the ALS that was described, but 20 years ago, there are almost no clinical trials for the brain phenotype of Williams. There are even very few symptomatic-driven treatments. So we conducted a retrospective study with methylphenidate on children with Williams syndrome and ADHD. It was effective, but one of the side effects of methylphenidate is that it makes the children more quiet, a little bit sad, and the parents who reported the side effects, the parent of children with Williams, they noticed the difference, and they didn't like it because they called their children the happy children, the children of love, and when they become more withdrawn, the parents don't like it. But the main emphasis here is that we don't have treatments for Williams. So one of the candidate genes for the brain phenotype in Williams syndrome, out of the 26 genes within the deletion region, is the GTF2I. And this is for two reasons. In humans that exhibit mutation within this gene, there are very rare cases with a mutation and deletions within this gene. We find hypersocial behavior, cognitive deficits similar to Williams syndrome. And there are also heterozygote knockout animal models that also exhibit similar behavior. So I mentioned Boaz Barak from Tel Aviv University, and when he was at Boston doing his postdoc, he created an animal model of this GTF2I knockout. He did a knockout, but the knockout was specific for the forebrain excitatory neurons, only for neurons of the forebrain. And what he found out that the knockout of this gene in the forebrain, change the expression of the genes that are in yellow. And these genes, 70% of them that were downregulated were related to myelination. So what he found out that what we know about myelin, and in the knockout mice of this myelin, of this model, that the axon velocity and intensity, which is affected by myelin, is decreased in these knockout mice. So that's affect connectivity of the brain, the problem with the myelin. And he also found out in the research of both mice and human tissue of postmortem of Williams syndrome that there is a problem with cells called oligodendrocytes. The oligodendrocytes are type of cells that are responsible for the creation of myelin in the brain. And the problem is that they have stem cells like the primitive cells that create the oligodendrocyte, the stem cells. The number of them is okay, but maturation to a mature oligodendrocytes, that's the problem. And another problem in the knockout mice that the association between the oligodendrocytes and neurons is impaired. 2 minutes, wow. Okay, so I'm going to skip that. And what we found out, and I'm going also to skip the model of the, he found that the white matter or myelin deficits that was found in these mice could be corrected with clemastine. Clemastine is the medication that we are going to try also in humans. And what the clemastine makes, that it increases the maturation of the oligodendrocyte cells. And by that, it increases the myelination in the mice brain. And he has shown that in the mice knockout model, after you administer clemastine, the white matter goes up, the motor deficits of the mice improves, and also the social behavior is normalized. What's the abnormal behavior of the mice? That if you put him in a cell and on the one side you have an object and on the other side you have a mice that is a stranger, the Williams will go to the stranger all the time and will ignore the object. It's kind of the opposite of what you see in mice model of autism. So the clemastine also normalized the hypersocial behavior of the mice. And what we are trying to do now, with the funding of Spark, and also with the help of Ran and Dana from Teva, we were struggling with the design. We are a single center. So it took us time to give up the hope that we will be able now to do a double-blind placebo study. So we are going for an open study. It's a repurposing of FDA approved because, as I said, clemastine is approved for allergy. It has also an antihistamininergic effect besides the brain effect. And we are going to now, we just, by the way, Ran, we just ordered the medication. So we are going now, we have to start in 30 days. It's going to be on 30 individuals with Williams syndrome. We want them to be young, but not too young because we need to be able to measure improvement. So it will be between 6 to 30. And then we consulted with the parents. What would be the most significant outcome from your point of view? And they said cognition. The mean IQ of individuals with Williams syndrome is within the mild range of cognitive, of developmental disability. They are independent. They cannot live as adults independently. Obviously, they don't have matriculation exams. They don't go to the army. So obviously, you need to improve their cognition and functioning. So the primary outcome measure in our study is going to be the cognition, and we use 2 batteries. One, the traditional one for IQ, which is the Wechsler intelligence scale, And the one that is newer and automatic, computerized, with also reaction time measures, is named the NIH Toolbox, which is also good for individuals with low IQ. The design would be an open study. We are going to have a baseline, a 3-month evaluation, and then after 6 months, another evaluation. We're also going to have, obviously, behavioral assessments, and the neurological outcome will be auditory brain response to see if really we also increase the nerve conduction by affecting, which it will be indirect measure of myelination. Hopefully, the results of the open study, we will be able to design an RCT, double-blind, but for that we will need additional centers for such a rare disease. Okay.